DNA, RNA, and Protein Synthesis. By Arya, Aubrey, Kaylee, and Nidhi. Part 1. DNA. DNA is a nucleic acid that carries and expresses an organism's genetic information found in the nucleus of a cell. It stands for deoxyribonucleic acid. DNA is constructed of 3 components: a 5-carbon sugar, which is deoxyribose in DNA, a phosphate group, and a nitrogenous base. Together, these monomers of DNA are called nucleotides. DNA is double-stranded with a double helix, ladder-like structure. The sugar-phosphate backbones form the ladder's sides, and the nitrogenous bases bonded with hydrogen bonds form the ladder's rungs. The 4 nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine. The rules of base pairing states that adenine always connects to thymine, and cytosine always connects to guanine. DNA also has antiparallel strands, so the ends of DNA are labeled with either 5' or 3' depending on the position of the carbon atoms in the sugar molecule. DNA replication. Right before a cell divides, it must duplicate its DNA in a replication process. This is during the S phase of the cell cycle. First, the 2 strands of DNA are separated, or unzipped, by an enzyme called helicase by breaking the weak hydrogen bonds between the base pairs, forming 2 replication forks in the DNA. Then, 2 new complementary strands are synthesized by another enzyme called DNA polymerase by following the rules of base pairing. DNA polymerase can only run in the 5' to 3' direction. So the leading strand is built continuously, while the lagging strand is synthesized in pieces that are ultimately stitched together. The process of DNA replication is semi-conservative because each DNA molecule resulting from replication has one of the original and one new strand. In eukaryotic cells, replication can begin at dozens or hundreds of places on the DNA molecule. Then it proceeds in both directions until each chromosome is copied. Part 2. RNA. RNA is another nucleic acid that is used to implement the genetic code. Unlike DNA, RNA uses the sugar ribose instead of deoxyribose, which means it has an additional oxygen. Its structure is also single-stranded, rather than double-stranded, and uses the nitrogenous base uracil in place of thymine. 3 main types of RNA are involved in protein synthesis. Each type of RNA has a different function. mRNA controls the assembly of amino acids into proteins. Proteins are constructed on ribosomes, tiny organelles composed of 2 subunits of several rRNA molecules. Lastly, tRNA transfers RNA. This RNA molecule carries amino acids to the ribosome and matches them to the coded mRNA message. Protein synthesis, transcription. Copying a base sequence from DNA to RNA is transcription. Transcription uses a vital enzyme called RNA polymerase. RNA polymerase binds itself to sections of DNA called promoters and separates the strands. Then it uses one strand of DNA as a template to assemble nucleotides into a complementary strand of RNA in the 5' to 3' direction. A cap is added to the 5' end of the molecule, and a poly-A tail is added to the 3' end. Before this new RNA molecule is ready, it needs to be revised and edited before it can be read, so it is called the pre-mRNA. Then, bits and pieces, called introns, are cut out. The remaining pieces are known as exons, which are spliced together to form the final mRNA. Protein synthesis, translation. The 4 bases of RNA form a language with 4 letters, A, U, C, and G, representing the nitrogenous bases. This language is called a genetic code, which reads 3 bases at a time, also known as a codon. Translation is the process of decoding or translating the mRNA genetic code to form polypeptides and eventually proteins. A ribosome attaches to the mRNA molecule in the cytoplasm. Next, the ribosome begins translating at the start codon, AUG, by attracting its anticodon, which is each tRNA molecule's group of 3 unpaired bases complementary to the mRNA's codon. The first amino acid is methionine because the start codon is always the same. Then the ribosome binds the next codon and its anticodon, and joins methionine to the amino acid brought by the next tRNA with a peptide bond. In this case it is lysine. This starts the chain of amino acids. After that, the ribosome keeps attaching amino acids to the chain, one at a time as each codon passes through the ribosome until the stop codon is reached. At this point the polypeptide is complete and it is released from the ribosome along with the mRNA. Part 3. Mutations. A mutation is an alteration in the base sequence of DNA, which can occur as a result of errors in the process of DNA replication, or by certain chemical or physical environments. Mutations can occur in one or several parts in DNA, called point mutations, by substitutions, insertions, and deletions, or even with whole sections of chromosomes. The factors that cause mutations are known as mutagens. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. Physical mutagens include some forms of electromagnetic radiation, such as ultraviolet light and X-rays. If these agents interact with DNA, they can produce mutations at high rates, and if the cells cannot repair the damage, the DNA base sequence permanently changes. Some of the most harmful mutations dramatically change protein structure or gene activity. For instance, certain cancers are the product of a mutation that causes the uncontrolled growth of the cell. Other mutations can be beneficial by allowing organisms to better adapt to their environment by producing proteins with new or altered functions. For example, some mutations increase resistance to HIV, the virus that causes AIDS. Additionally, in humans, beneficial mutations can give people greater bone density and strength. Finally, most mutations end up having little to no effect on gene expression at all.